Hello and good morning, everyone. I'm very excited to see so many of you in the new round of the lecture series of the Single Cell Omics Network in Germany. So today we have Laleh Haghverdi presenting her work. And before I introduce Laleh, I would like to give a bit of an organizational thing. So please stay tuned for our next lecture series in 2 weeks, where we have Christian Schmiedl presenting from the Leibniz Institute for Immunotherapy in Regensburg. So that's on 6th May on the same channel. So please do come to this talk too. Furthermore, please save the date for our SCOG workshop for about computational single-cell genomics in Munich in September 12th to 13th. And it's going to be an in-person workshop again. So we're super excited about that. And we're still working on the speaker lineup. So please stay tuned for updates. So the lecture today from Laleh will be 30-minute presentation and then followed by a 15-minute moderated discussion. When you have a question, please use the raise hand tool in the Zoom chat to indicate that you would like to ask a question during the discussion. If you're not feeling like talking directly, and unmute and share your video, you can also type it in the chat. And then I will read out the question. So with that, without further ado, we'll have Laleh Haghverdi today, and I would like to give you a brief overview on her vita. So Laleh studied physics for a bachelor in the Sharif University of Technology in Iran. And then she moved to do her master's in Germany, in Cologne. And after that, she started doing her PhD at the Technical University in Munich at the lab of Fabian Theis at the Helmholtz Center in Munich. And there she was really one of the pioneers in the single-cell computational biology. And after that, she moved for a postdoc at the EMBL EBI in Kingston, UK at the John Marioni Group to develop one of the, let's say, most widely used batch effect correction tools, MNN. And after that, she started her own group as the group leader for computational methodologies and omic analytics at the Berlin Institute for Medical Systems Biology, BIMSB, and the Max-Delbrück Center for Molecular Medicine. And she started that in 2020, and I'm totally excited to see what she's now doing in her own group. Laleh, stage is yours.
Okay, thank you, Maren, very much for the introduction, and good morning, everyone, to this talk. I am very happy to be here today with you and present, as Maren said, so the fair is like some work from my own group, so that we developed now it's getting almost 2 years, but yeah, so this is the first preprint and the kind of more or less finished work that we have, and it is about quantification of cell state velocities. So I start with a little background of what cell state velocities are. Next slide, please. Yes, so we are in general analyzing single cell data, and with single cell data, one of the interests is, of course, seeing the heterogeneity in the biological systems among the single cells, and studying dynamical systems like, for example, development, or when you have a treatment or anything that changes over time. So previously there were some methods developed for studying the dynamics part of systems with single cell data, like single cell RNA-seq data, and that was at the pseudo time. analysis. Basically, when you know where your pluripotent cells are, you can order the cells in a way to get to know that cells should be moving away from this pluripotent state towards more differentiated states, right? And then that's what there are different methods of calculating pseudotime, but that's the basic concept behind it, that it shows you the overall direction of the movement or development or whatever the dynamics is in your system. But the thing is, with pseudotime, we cannot capture the stochasticity among how the cells are moving. And for example, if there are cell plasticities, like from one position, they can move into two different positions. Or if, for example, they give different cells have a heterogeneous response to something like drugs. Or for example, can all the cells go a little back in the pseudotime, or is it always forward, right? So these are the kind of questions that we could not answer with pseudotime. Next slide, please. And for a new way to look at these things more efficiently that pseudotime was not able to. There was this nice RNA velocity paper from Lamano and his colleagues, that they suggested that by measuring two different stages of gene regulation, so namely the unspliced and spliced mRNA, you can add a dimension of time to your snapshot RNA-seq data, right? And with this new dimension of time, they suggested that so you have the transcription rate, which is alpha, then you get unspliced, and then there is splicing rate beta. Then you get the spliced mRNA, and then it can degrade with a gamma rate. And you can write a set of ordinary differential equations for this process. So for each gene, right? And by plotting your spliced versus unspliced counts, you can try to estimate these parameters and learn the velocity. So the velocity is just the derivative of the spliced counts with respect to time. So, and then once you get the velocity from every gene, you can sum up these vectors to get the high dimensional velocity for a cell. So in the original RNA velocity paper, they used a steady state model. So they were fitting a line basically to where the maximum measurement of S and U is lying. And then there was another nice paper in 2020 from Bergen and the colleagues that they suggested a more advanced parameter estimation method for this, that you fit the whole trajectory, not only the steady state. Right. Next slide, please. Okay, so there are maybe the first thing that we should notice about RNA velocity is that this model is basically composed, or it is assumed for smooth dynamics, right? So basically the solution of the ODEs with respect to time, like how the S and U curves will look like over time, are shown in this blue and red curves, and you see this is explaining smooth dynamics, right? So, and the paper in fact starts with an example from bulk RNA experiment that explains the circadian cycle in a mouse liver, and they could show that for this bulk data, for when you look at the phase portrait, so meaning the spliced versus unspliced counts for this data, you see this nice almond shape trajectories, right? And then they use their parameter inference method, and then they can show that you can infer which way each bulk sample is moving towards the next time point of the measurement. Next slide, please. However, so we should notice that for single cells, things are much more noisy, right? So first of all, we know that all genes expression, it is not very smooth, right? So, and once you have noisy dynamics, your average velocity will not be the same as your instantaneous velocity, because things are just going up and down, right? And another thing with single cell data is that spliced and unspliced measurements are very inaccurate, right? So as a result, so we have to do a lot of data smoothing to be able to fit some kind of almond shape that this kind of theory is telling us. Yeah, so, and when you do this data smoothing, then it is a little bit counter purpose, because our purpose was to see the stochasticity of the velocities, right? So among single cells, and the first step you start with is data smoothing, but I mean, you have to do it because the data is too noisy here. Next slide, please. Okay, so yeah, with that kind of introduction, I give an overview of my presentation today, but you will see later in my talk. And it is that we show that what we look for is to be faithful to the true signal and cell-to-cell variety of the cell state velocities, right? And we show that there are several problems in existing methods regarding pre-processing, visualization, and estimation of the velocities that actually don't let us to get the thing that we want to get from this data. And a very important point, again, is, as I said, so this cell-to-cell variety, right? And then we question that, like in previous methods, the agreement of the velocities with pseudo time was kind of used as validation. So yes, you can do that, right? But if you know the pluripotent state in your data, then you, as I said, want to get more from this kind of analysis, right? So, and we say, okay, maybe it's not good, so because pseudo time is only giving you the overall directions. And we come up with two different methods to address some of the existing issues. The first one is Kappa-velo, which is a detailed analysis, and it is building up on the previous methods. So the first method was a velocycle, and the next development with the dynamical model, SC-velo. So our Kappa-velo is just building up on SC-velo and taking it further, addressing some of the issues, but not completely. And then we have another minimal method, which is eco-velo. And it is a very simple method. It uses a little bit of more assumptions, but it can instead bypass a lot of cumbersome pre-processing and parameter estimation steps, right? And we think with the current state of the thing, like all the problems that we have with this smoothing, pre-processing, parameter estimation, this is actually a good and competent method in the current state to be used, that I will show you later. So next slide, please. Okay, so as I said, there is also some problems with the current visualization approaches. And here we show that a simulation that we made in such a way that it's a trajectory of moving cells, like a development or whatever. And we simulate it in such a way that we know in the beginning there is a lot of noise in the velocity directions, right? So that's how we simulate the data. And towards the end of the trajectories, the velocities become smoother and more directed in the same way. And we compare the true velocity. So this is only the visualization of the true velocities that we have in our simulation with a PCA projection and how SCOG, for example, is visualized in these velocity vectors. And we clearly see that SCOG's visualization is a kind of omitting or eliminating the heterogeneity in the beginning of the path, right? And the way that SCOG visualizes, and it is the same for Velocito as well. So I'm writing the formula for showing the displacement. So if your y is initial, is the beginning of the arrow, delta y is the end of your arrow, right? And both Velocito and SCOG use the same formula, and Pij is the probability of moving from one cell to one of its neighbors. That is calculated based on the velocity vector, right? So the only difference between SCOG and Velocito is how they calculate this PE matrix. But in the end, they see how does that P matrix project on every nearest neighbor of a cell. So Yi and Yj are all the nearest neighbors of a cell I. And they sum, they make a summation of all these different directions. So things to note is that this is giving you an expectation value where your next step will be, right? So, and this is again another level of smoothing your data, right? So when you talk about expectation values. So actually this kind of summation is not orthonormal, right? So because your nearest neighbors in the low dimensional space, they are correlated. So this is not a vector summation, but it is expectation value. As I said, it is smoothing things. And it depends, it projects the velocity vector only on the nearest neighbors of a cell. Therefore, it depends on what neighborhood you take into account. And in the end, it is changing the direction and norm of the velocity vectors, right? So we suggest in the next slide that we can use the Nystrom projection method for nonlinear, for whatever nonlinear data embedding that you use, for example, TSNE or UMAP, or for diffusion map, you can also do that, right? So, and then what the Nystrom method is telling is, so the first part, for example, is how the first row, we are showing what, for example, TSNE or UMAP or other methods are doing. So you start with a matrix, with your data matrix of N1 cells and G genes. Then you calculate a P transition matrix among your cells, and then you embed them in a low K dimensional, for example, 2-dimensional space, which is your Y. And the Nystrom method just says, if I have new data here, my original data would be my initial S counts, and my test data set will be end of the arrows, right? So I want to project end of the velocity arrows onto an embedding of the initial state of each arrow. So for my test set, which is here, actually my N1 and N2 are the same, but in general, it doesn't have to be the same number. So I can calculate a transition matrix between my new points to old points, right? And then I know how to project the old points into that embedding that I have, right? So, and then for the new points, I can just do P2 times the embedding that I have, right? So it is just, as I said, like projecting N2 to N1 and then N1 to K. And here you see, for example, for this simulation, with this TSNE projection, we do capture that heterogeneity in the beginning and more aligned velocities in the end. So this, was with the visualization. Now we can go to the next slide, which is another issue that is important in the RNA velocity formalism, and that's the scale invariance of velocity components. And that means that if you solve for the ODE equations for a gene G and you get alpha, beta, gamma as solutions, then kappa times these parameters will also be a solution, right? And this will be a problem when you are combining the velocities from multiple genes in a vector sum. So again, we are showing in this example, for example, your velocities without the kappa scaling for each gene. So for gene 1 and gene 2 could be shown by V1 and V2. And then when you scale V1 with the kappa 1 and you scale V2 with another kappa 2, you can see that it can completely change the direction and the length of your velocity vector, right? So to solve this scale invariance issue, what Velocito was doing, it was assuming that the splicing rate is the same for all genes, right? So to actually omit this degree of freedom that we have. And what SCOG does instead is that they initialize an expectation maximization and algorithm for each gene. They assume that the full cycle time is the same for all genes, right? And we thought maybe none of these two assumptions are very realistic in a biological system. And then we propose something else in the next slide, which is based on the cell densities. So we say that when we collect RNA-seq experiments, the probability of capturing a cell in a given state is proportional to the time that the cell was spending in that state, right? So if the cell is going fast, the probability that you capture a cell in your experiment is lower. So this is our assumption that we think it is a bit more realistic, maybe from compared to the other two. And of course, I mean, it can be not the case if you select and enrich for specific cell types and these things, right? So, but still, in a lot of RNA-seq experiments, we see this, that in fast-moving parts, there are less cells captured, right? So, and then we also ignore that the cell birth and death rate can be different in different parts of the trajectory. So what we do is we just use the solution for the unspliced count, and we just shuffle the things a little bit too. And then instead of the delta T between two cells, for each gene, we use the number of cells that we capture between them. Can we go to the next slide? Yes, and then, we can get the kappa for each gene as the slope of the right-hand side over delta T plot. And if we have a lot of cells in that top right part, which is the steady state, then we don't get only a line, but we fit a parallelogram, right? And then still we can use one side of it, the slope of the line, on one side of it as the kappa for each gene. Next slide, please. Okay, and then we validate this method of estimating the different timescales for different genes. So on the simulation data, where we show how the true kappa we get is correlated with the fitted kappa, and the velocity components. Also, for us, it has a higher correlation compared to SC-velo, and then we also compare the cosine similarity, like the angle between the true vector and the inferred vector and its norm. So on the bottom, we are showing the plots in the PCA space, and on the top, we are showing it in the high dimensional space, always comparing SC-velo with the Kappa-velo results. Okay, next slide, please. And now we come to real data, so which is a pancreatic endocrinogenesis data set. Right, so here we show that the velocities, so both on the inference step and visualization step, so we see the Kappa-velo results. And so when we use Kappa-velo visualization, we see that, for example, SC-velo is getting sometimes too long, like, I mean, a little bit of inconsistencies in the velocity vectors. And for example, in the beginning also of the path on the right, so which is about the cell cycle, the arrows should be longer because cell cycle is faster thing, right? So, and basically you don't see these kind of differences in the Kappa-velo estimated velocities. But when we use SC-velo's visualization, because all the velocity vectors are only projected on your nearest neighbors, you are like killing some of that true signal that was coming from the analysis, right? So, and then we also show that with our method, we get a more reasonable range for the splicing rate compared to Kappa-velo, so which is 10 to the minus 1 to 10 versus for 10 to the 4 times magnitude of variation for SC-velo. And yeah, we are showing some examples of fast and slow genes depending on how many cells were captured during this cycling time. Next slide, please. Okay, so this was with the Kappa-velo, and now I come to the eco-velo, which is like a minimal model as I described earlier. So this, the idea actually came also from the Lamano paper, the circadian cycle in mouse, where they showed that the unspliced counts at a given time are similar to the spliced count in the next time step for the bulk data, right? And we thought how this observation could, how we can describe this observation in terms of the ODE model, right? And we just wrote the velocities, like as we get the from the ODE, and then we look the S at the splice count for a gene at T plus delta T. It will be like this, and then yeah, just it will be something times the current splice count plus something times the unspliced counts times the delta T between the two measurements. And now if we set fixed delta T to be 1 over gamma, so that's the time that you need to degrade all of your existing spliced RNA, mRNA, right? So as a result, your first term in that equation will become 0. So there, and your S of t plus delta t will be proportional to the unspliced counts in the time step before u of t, right? And now if we also assume that beta is constant between all the genes, we exactly get S, right? So if we can assume that beta and gamma is the same for all genes, then S equals U is basically what you get, right? So, if you remember, Velocito was assuming that beta is the same for all the genes. Now we add one more thing. So we also assume that gamma is the same for all the genes, right? And suddenly we have a super easy model. And in the next slide, that we can just look, that we can just say the beginning of arrow is S, and the end of the vector arrow is the U, that is the closest nearest neighbor to that S, right? So we find basically, we identify mutual nearest neighbors between the S counts and U counts, and then we direct the arrows from, from U to S. I confused a little bit. But yeah, here you see the this eco-velo for the simulation. Again, we do capture that heterogeneity in the beginning and more aligned vectors in the end. And on the real data, you can see that we get quite sensible velocities as well, right? Despite the simplicity of the method, with just adding one more assumption. Okay, so maybe that's not very realistic. We know that different genes can degrade on different rates and all of that, of the same for a splicing rate. But given all the problems that we have for parameter fitting, data smoothing, and all of these things, maybe we think this can also be a useful method if we select maybe genes that their splicing and degradation rates are a bit more similar, or somewhere to see if this assumption is more or less good for your data. You can use this method as well. Okay, next slide, please. Yeah, so yeah, we actually describe the pipeline for both of our methods, and then there is actually more in the paper that I didn't talk about it here because of time mainly, and especially for pre-processing, right? So we fixed a few steps in the pre-processing of the data, which also improves the results. For example, we can omit a lot of Merck genes, like genes with multiple kinetics, and that were reported in previous studies. We show that many of them were just an artifact of the inconsistent pre-processing. And yeah, as I said, we revised the visualization and also scaling of different velocity components. And then, yeah, the pipeline for eco-velo is there as well. Next slide, please. Yes, so yeah, you could, of course, if you are interested, you could check our bioarchive paper, and the link for the GitHub, it is also in the paper. So things are, it is usable functions for now, right? So, but we decided to make the package for when we are going to really publish the paper. So in our revision time or something. So, but definitely there are the existing functions are usable now, and you can try them. And yeah, if you have questions, ask. Feel free to contact Valerie and Brigitte. So you can find the contacts are on our webpage. And so from there, this is going. as I said, we try to improve the existing methods a little bit, but of course there is still way more to go, right? And these inaccurate S and U measurements are a fundamental problem that I started the presentation with, and then parameter estimation can be maybe improved. And because of this intrinsic difficulty in spliced, unspliced measurements quantification, maybe it is also a good idea to look for coupling other pairs of omic stages to get this time, this temporal dimension in the data, right? So, and then when you use the different types of omics, then it means that you are looking at different time scales. Right? So for example, for spliced and unspliced, maybe the difference is you're looking at the average scale, average velocities in a shorter time scale than, for example, if you would combine mRNA with protein level, right? So that these are average velocities in different time scales, and that would be also interesting to know what is the short-term plan of the cell, like maybe more noisy one, and what is the more long-term plans, depending on what time scale you are considering. Okay, the last slide. And yeah, the last slide should be acknowledgement. No, the one before. Yes, yeah. Thank you. So yeah, so this work was done by Valerie, Brigitte, Fergal, especially Valerie and Brigitte, who are the first author on our paper. And as I said, just you can find our contacts on our webpage. And I would also like to thank my other group members, Carla, Ehsan, Subhana, and Sid, and the funding institutes. And thank you very much for your attention.
Yes, thank you, Laleh, for this very math-heavy talk, but very interesting and also important question about RNA velocity. So I agree that we still have a long way to go to get good RNA velocity and estimates. And I would like to open the discussion. So folks, please raise your hand if you like to ask questions to Laleh directly. I'm trying to see whether people are raising their hands. So there is Piyush Yoshi. So please unmute, please probably also show your camera or activate your camera. My
camera is not working for now, but
no worries.
I also put the question in the chat. I hope you can hear me. So I think the talk was very nice, but one of the things which well, there are sort of two questions. The first is that now we are actually using single nuclei RNA-seq data a lot of the time. So we don't have this correct estimation of unspliced versus spliced, because most of the nuclei transcripts are unspliced. So what would be a way to understand cell state, you know, the thing that you're doing with the single cell data in a single nuclei? Okay, and the second is that even if we use this single cell data, how are you going to sort of in a large data set where we have, you know, when we are making a cell atlas, we have sort of multiple time points, and in some of the samples, there's a lot of changes that are happening, but as we go for the adult samples, you know, the changes are not so much. So how are you going to integrate the data and then define these velocities in an integrated data? I don't know if my question was meaningful.
Yes, so for that first part, I mean, I don't think you can do something, right? So, right. So that, as I said, like you could do pseudo time. You can use that pseudo time analysis just to get the overall direction of how the cells are moving, right? But as I said, for calculating cell state velocities, you need measurement from two different stages, spliced and unspliced, right? So, and if you say in my data, I don't have any spliced counts, okay, so you don't have that temporal dimensions. There is not much that you can do. And for the second question, if I understood it correctly, you are telling how in combined batches of data you can estimate velocity.
Yes.
And tell that we currently there are a lot of problems with like cleaning the data and pre-processing, and you are just making it more complicated again. I think currently maybe things are not very promising for the data from multiple batches, right? So because always what you need to have is this S versus U for different count for different cells, right? So that you fit this almond shape too. And yeah, once you have data from multiple batches, okay, you can do batch correction, but we know that batch correction methods are still, they leave usually some batch effects there. So it is really difficult, and I don't know.
Would you then, sorry for taking more time, but would you then calculate for individual samples and then sort of project that onto the diffusion map? Because you can do diffusion map for an integrated data and sort of identify, you know, how the trajectory is going, but you have calculated these velocity vector for individual, like the cell-to-cell transition in individual samples, and sort of somehow project that on that integrated diffusion map, just as a thought.
Yes, I mean, actually, maybe then I would say maybe eco-velo is more hopeful for this, but actually we tried a little with data from multiple batches and we saw the difficulties there. So again, this has to be studied more. I cannot answer too much.
Yeah, maybe we move on to Mo's question.
Yeah.
So.
Yeah. Hey, Laleh, that was great discussion, also talk. I have a question regarding the applicability of like velocity models, because we know that like I think it's just like if you dig into like papers that can use velocity, then you can find a lot of wrong conclusions because of the robustness of all the problems that you mentioned. For example, like still not clear why, I mean, we know that the different genes follow different dynamics, so probably more complicated models are needed. So it's like a fixed assumption for like all genes would not be enough. But like this Kappa method that you showed, I was wondering whether you can use those Kappa values, for example, to point out when the model will fail. For example, if you have like transcriptional bursts, because you have this notion of fast and slow genes, to just show where the model basically fails, like or any hint on like when the model basically fails on which data sets.
I mean, if we can capture the kappas, then we can estimate better velocities, right? So I mean, we cannot use that thing to say when we fail. Maybe we can use Kappa-velo to say when SC-velo was failing, right? So
yeah, exactly. Yeah. For example, for hematopoiesis, or like when you have like faster or like slower processes where then the counts will not be captured properly. Yeah.
Yes. Yeah. We showed this actually, right? So in the on the pancreatic data, we show like, for example, there are a group of fast genes and a group of slow genes, whereas SC-velo is basically assuming the same cycle time for all genes, right? So that's unrealistic. And then like the single cell velocities, then yeah, we do compare them.
I see. Okay, thanks.
Thank you very much. Denise, Duma, would you like to maybe activate your camera and ask a question?
Sorry, I cannot use it. So I just wanted to ask if you can, I mean, you'd already discussed this, but if you can explain a bit more how it's compatible, the time scale of these RNA velocities with developmental time scales like this pancreatic embryogenesis. So it seems to me like these are very different times. So how can you use them together? I mean, to infer, if you can explain a bit, because to me it seems like there's a gap in terms of timescales. So is this the right modality to study development?
So I think for the pancreas data, like the beginning part, which is cell cycle, it is on the order of hours, but like the whole differentiation towards like delta cells, like at the end of the trajectories, it is, if I remember correctly, on the order of 20 hours or days. No, I don't know. Can someone help me? Maybe in the chat.
Maybe I can jump in. So the cell cycle in the beginning, it's a little less than a day, and then the differentiation process along the trajectory in the pancreas example, it's I think 4 days that it takes from beginning to end in this sample.
Right. Yes. And the thing is that we capture different cells all along these trajectories, right? So, now the thing, like the time scale between spliced and unspliced trees, it can be also on the order of minutes to hours, right? So the velocities that we get is like, it is not over days probably. So it is like the velocities, average velocities over a time of a few hours maximally, right? So, but it is like telling us this cell is moving this way, right? So, and we have different cells sampled along all this trajectory. So we don't need to capture the whole time scale of the whole system, but like in each part, we say like an average velocity over a few hours is this direction or that direction, and that should be fine.
Okay, I see. Okay, thank you.
Okay, thank you. So one last question from the chat. So Carlos Ramirez asked, is it possible to accurately measure changes in mean velocities under different conditions? So for example, if we know that our cells differentiate faster under some condition, do you think it is possible to measure these changes using the available velocity methods?
I mean, we get some sense of how fast our gene is moving, right? So through these kappas that we estimate by the number of cells that were captured between 2 steps, right? But yeah, I would say like the current methods, they are not really well tailored for considering the time scale over which that we are reporting the average. And that is actually one of the points that we discuss in the paper in detail, that this average time matters. What time are you talking about? And in fact, because alpha, beta, gamma are different for all different genes, so we are combining kind of velocities with different, maybe not a lot, right? So all of them are on the order of hours, but minutes to hours. But again, still we are mixing different time scales, and the current methods are not. But for example, what we do with eco-velo, so there we really fix the delta T for all genes, assuming the same beta and gamma. So in that respect, maybe it is better respected, like what that average velocity means.
Okay, thanks. I think this is like still a super interesting question, whether we actually can detect, let's say, blocks in differentiation for knockout models or so on. Lots of say development still to be done. And with that, I would like to close the discussion. Mauricio, I'm sorry that I cannot come to your question anymore. So thanks all for participating and discussing so lively. And with that, I thank Laleh again for her talk, and see you all in 2 weeks for our next lecture series.
Thanks for the discussions and attention. Bye, guys.